Great. Thank you for the introduction. Yeah, so the question comes up often, why I talk about genetics. I've been fortunate to be involved with a number of large sequencing projects in the United States, and I think we've really redefined the genetic basis of congenital heart disease over the past 5 years. You know, when I first came up with the topic to talk about genetics and surgical outcomes, I was discussing it with my colleagues, and I quickly came to the realization that perhaps the better topic would be to just talk about some of the basics of genetics. And because I think there's some important, very simple points that I could pass on, and it would, and I think it's things that could help you clinically. For instance, one of the most common questions, and I'm sure you get it also, is you have parents come to you who have a feeling of guilt and ask you, the congenital heart disease that my child has, was this from me? Did they inherit this heart disease from me? And questions like that, I think you can answer. And I hope by giving you some basic information, I think you could, it could be useful to you. So the basics of genetics as we know it these days started about 15 years ago with the sequencing of the human genome. That's the Human Genome Project. And I think it's actually remarkable that today, 15 years later, you can actually, in the United States and worldwide, you can spit in a cup, you can send it off, and then they can send you back some information about your past and some of your proclivities and some of the ideas that your genetics can inform you, about you. And I think it's a remarkable advance. But it's not everything, certainly. So for this lecture, I'm going to really talk about 3 things. First, I'm going to talk about some of the basics of the human genome. I'm going to talk about some of the genetic variants or mutations that lead to CHD. I use the word variants because, yes, they are a form of mutation, but mutation has really a negative connotation that, but as I'll discuss, it really is the basis of all human individuality. So it's not really a mutation per se, but really a variation. I'm going to discuss the dominant mechanisms behind why or how these genes work, and it will give you some insight into what complications or what other associated features in addition to congenital heart disease you can expect from some of these mutations. And then I'm going to talk about some of the specific genes. And then I'm going to talk about inheritance and recurrence and some basics if you don't have the ability to do genetic testing, which are based upon population studies. And then if you do have the ability to get some genetic testing, who and what you should get. So what are the basics? So human genetic diversity is based upon differences in your genetic code. And your genetic code is based upon 3 billion base pairs. And the variation basically comes in 3 sizes: big, medium, and small. That's like all you have to remember. Big is chromosomes. We obviously have 23 pairs of chromosomes, and you can have problems with these chromosomes. Then there are medium-sized differences, which are what we call copy number variations, which are either duplications of small areas of DNA or deletions of small areas of DNA. And these are range in size from a few hundred base pairs to a few million base pairs. And then there's very small variations that are single nucleotide changes. And there's tons of them. There's over 4 million that are found. And this is the statement. It's that individual variants are populationally common, but the combination of variants is individually unique. So I'm going to explain that to you right now, because I'm sure no one got that. It's hard to understand. So here it is if you have an individual genetic change, right, it is likely that if there's 100 people in the room, that somebody else in the room will have that same exact mutation, right, or same exact variant, okay? And overall, there are going to be a group of genetic changes or group of variants that are likely shared by many people in the room, right? That's like some of the racial genetic variants that you have. That's why, for instance, Koreans in general are very good looking and smart. No, it's just joking, just factoring. Okay, and then, but the sum total, right? What makes you unique is that the variants that are shared individually that are shared by other people, when you have all the combinations that are in you, that's very unique. No one else has your particular combination of variants, even though they may have one or two of them, or they may have 100 of them. Altogether, the combination of them is yours and yours alone. But these shared variants, both the small, medium, and large shared variants, these are called common. And the definition of common is greater than 1%. That mutation or variant is shared by at least 1% of the population in general. Doesn't sound like a lot, 1%, but it is. So 1% is common, it's not rare. Rare is much, much less, okay? So it was, and this is important, and I bring up the difference between common and rare because it has implications for the studies that we've done in the past and the studies that we do now, and also about inheritance, okay? Originally, maybe 5 to 10 years ago, you may have heard about GWAS studies, genome-wide association studies. And these were based on SNPs. You may have heard also about SNPs, single nucleotide polymorphisms, small little changes that you could test on people. And people said, oh, okay, I'm going to do a SNP chip on everybody. I'm going to look at your SNPs on everybody, and I'm going to find out what SNPs are associated with congenital heart disease, right? But it was flawed because SNPs are common variants. In other words, they are found commonly among the population. And what we have found that common variants do not cause congenital heart disease. So when all these GWAS studies, they were found and associated with many diseases, but as a rule, common variation that is the most common that we share amongst each other, this forms your background variation. It does not cause congenital heart disease. What causes congenital heart disease? Very, very rare mutations. These occur in less than 1% of the population. They are highly damaging, and they are likely to be de novo. What does de novo mean? De novo means that your parents don't have it. The child has it, but the parents don't have it. So why is this important? Okay, so if there's going to be one takeaway lesson, I think, of the whole thing, it's that in general, if your family, the family of your parents, the parents of the child who has congenital heart disease, in general, not always, but in general, if there is no history of congenital heart disease in their family, the child did not inherit congenital heart disease from the parents. It happened new. Now, why is this the case? Like, why would it seem like that's the case? It makes a lot of sense genetically. And that's because up until the most recent generation, which is now because everybody is operating and doing things, if you had congenital heart disease, you died. So unlike other disease entities, the genes, the adverse genes, did not have an opportunity to build up in a family, right? So you never accumulated adverse genes, because if you had a gene mutation that caused congenital heart disease, you died. You never reproduced. You never passed those on. So unlike other families, for instance, that had these big pedigrees of disease where people found, congenital heart disease never developed that way. So uniformly, if you have, if a child has it and there's no family history, it's likely de novo. It's a spontaneous new mutation, okay? And causative congenital heart disease up until this recent generation are fatal. There are some predictable exceptions, right? And I'll go into them slightly later. There are probably some things where there is some family effects, and I'll tell you later how it could be predictable. And it basically has to do with sometimes you can have a mutation and it's not expressed, right? It's called incomplete penetrance, or you may have a lower phenotype like a bicuspid aortic valve, and then you can pass those on and it can be expressed to a higher degree in the children. But they're predictable. You understand why LVOTO may have a slightly higher risk. Okay, so basically, I'm going to summarize a little bit. Your genetics and the genetics of congenital heart disease are based into causative and non-causative variation. The causative variants are very rare, usually less than 1%. And again, as I said, there are those big, medium, and small changes. There are big, medium, and small causative, and there's big, medium, and small non-causative, okay? But as I will kind of describe later, some of the nuances is that for congenital heart disease and causative congenital heart disease, many different gene variants can cause the same problem, right? So in other words, there's a gene mutation that can cause tetralogy of Fallot, and there's another gene mutation that can cause the exact same tetralogy of Fallot. Okay, that's called genetic heterogeneity. Now, what about your background variation? All these genetic changes that you have harbored in you but are not pathologic. They don't cause congenital heart disease. But can they affect congenital heart disease? Yes, right? So not everybody who has a causative mutation gets the disease, right? And that's called incomplete penetrance, right? So and of course, not all congenital heart disease is genetic, right? There's rubella, there's exposures to other things that we don't know, right? So not all congenital heart disease, but if it's genetic, just because you have the mutation doesn't mean that you will get it. The other thing is that your background variation can affect how the disease is expressed, right? So the same mutation in one person may have significant PS. It may, in a different person, have pulmonary atresia. So your background genetics can affect how the disease is expressed. And finally, there are genetic variants that can affect how you handle congenital heart disease, but may have nothing to do with the heart. For instance, you know, there may be there are certain gene mutations that will change the way you metabolize drugs. And so you may be, or for instance, aspirin resistance. These are important genetic variants that don't directly have an effect on congenital heart disease, but certainly have an effect on therapy. So all of these things kind of contribute. Okay, let me give you an example. One of the earliest kind of gene mutations to be found was NKX 2.5. And this is important because it explains many different things. For instance, if you have an NKX 2.5 mutation, some people have no structural heart problem, but they have arrhythmias, right? So it can manifest as an arrhythmias. Some people have atrial septal defects. Some people have hypoplastic left heart syndrome. You would not even expect them, you know, you may expect some left-sided lesions to group together, but that isn't always necessarily the case, and NKX 2.5 is a great example. In general, I'm just going to summarize. There are the big lesions, chromosomal aneuploidies. Those are chromosomal number problems. You know what kind of lesions these are. It's like trisomy 21. It's like Turner syndrome XO, right? These are examples of chromosomal aneuploidies, and they contribute about to 10% of the known genetic causes of congenital heart disease. Then about another 10%, it's 10%, 10%, 10%. Another 10% is these medium-sized copy number variants. Examples of a copy number variant would be DeGeorge syndrome. Also, a copy number variant is Williams, is another example of a copy number variant lesion. And then the single nucleotide variants that are associated with the disease, we predict there's going to be about 400 of them, but we only know about 200 or so of them. Some things are like ZIC3 with heterotaxi, and KX2.5 is another example. So small, medium, and large, 10% each. So how do the variants cause disease? So we know now that there is genetic variation. There's both causative and non-causative variation. So how do these variants actually cause disease? So if you look here on the left-hand side, the top is an example of a stretch of DNA that leads to expression of a gene. And in the middle at 598, there's a break point. In other words, there's a mutation at that point. And it could lead to many different truncated forms of the disease, or truncated forms of the gene. In some diseases, this truncated form works as a dominant negative. That means that it goes and it actively represses a protein. It actively represses some system, okay? Another thing that can happen, particularly in cancers, is a gain of function. In other words, the mutation will lead to increased activity of something, okay? Congenital heart disease, that doesn't happen. It's very simple. In congenital heart disease, overwhelmingly, if you have a mutation, it leads to decreased expression of the gene, haploinsufficiency, okay? So that's generally speaking how it happens. And it's also very interesting what we've learned is if I had asked people 10 or 15 years ago, I said, if you have a heart problem and you had a gene mutation that led to the heart problem, what kind of genes would be affected? And some people would say, okay, structural heart genes, genes that make up the heart muscle, genes that make up the heart valves. That is not true, okay? So what we found is that the mutations occur in genes that occur in either cell signaling, such as TGF-beta, cytokine signaling, regulatory networks that affect the folding of the heart, okay? Folding, organization, heart development in that fashion. And it is not a structural heart gene. And actually, when you break this down, this makes, again, a lot of sense. Because, for instance, when the heart is developing, it relies on a TGF-beta, which is a hormone or a growth factor, is a great example. The heart requires TGF-beta to develop normally, but so does the brain. So when you have a mutation that leads to a problem in TGF-beta that led to your heart defect, guess what? That same problem exists in the brain. So these kids, for instance, are prone to neurodevelopmental problems also. So it makes perfect sense why when you have a heart problem, you have many other associated problems. So that's number one. Number two is, remember I said that there's genetic heterogeneity. In other words, that many different genes can lead to the same problem. And that's because when you affect regulatory networks, all these genes in the middle are involved in the regulatory network. And if you have a mutation in this gene and you have a mutation in this gene, they may create the same problem, even though they're two separate genes, okay? So the fact that the mutations were found in these cell signaling pathways and these cytokine pathways is very important and really helps us to understand some of the other problems associated with congenital heart disease. Okay, so that's basically the background of, and really all you need to know. But if you want to ask some practical questions, you can start with this one. Is all congenital heart disease inherited from your parents? And generally speaking, it's not, okay? So if you ask the family, is there any other family history in your family, that you have to ask that question. Because if there is, it's all bets off. Generally speaking, it's not. But you can do better if you have genetic testing. So let me explain. So first of all, if you don't have the capability to do genetic testing, which is very expensive, there are population studies. And when people talk about recurrence, there's two different recurrences, right? So if a kid comes in and has congenital heart disease, one question that people have is, if I have another child, what is the chance that I'm going to have another kid with congenital heart disease? If you look at population studies, it's about 3% is the chance that you're going to have another kid. But if you have genetic testing, you can actually do better. You could have even a better idea, and I'll explain that in a second. The other recurrence question that you can get asked is so now I have a child who has congenital heart disease. What is the likelihood that that child will have a baby who has congenital heart disease, right? So that's a different kind of recurrence. And that answer is 5 to 10%, but it can be much different depending. By the way, it's a difference if your father has a congenital heart disease. In other words, if the child is a boy, the chances that he will have a child who has congenital heart disease is around 5%. If the child is a girl, the chance is around 10%. So there is definitely a sex-related difference, and basically, if the affected person is a mother, then it's higher. Okay, so and there are some specific instances, as we spoke about before, that left ventricular outflow tract is probably higher, and that's because there are these unrecognized forms of left ventricular outflow tract obstruction, mainly bicuspid aortic valve, that potentially could be higher, but not all studies are in agreement, okay? The other important thing when you read genetic studies is, for instance, the largest genetic study talks about relative risk and absolute risk. So for instance, if you look at the largest population studies that were done that are Danish studies, the relative risk, if you have a child with heterotaxy, the relative risk of having a recurrence is 80-fold higher. And you say, oh my gosh, that's crazy. But the absolute risk is still only a few %, right? And that's because the risk of having heterotaxy is so low anyway. So it's 80-fold higher of a very low risk, right? So your absolute risk and your relative risk are completely different. Okay, heterotaxy, as I said, many people believe that heterotaxy is highly heritable, but it really hasn't been borne out, but the risk is probably 5 to 6%, okay? So here is where you could do much better, okay? So what is a de novo dominant mutation, right? So generally speaking, de novo means that it's not inherited. If you know for a fact that the mutation that exists in the child is not inherited, and the only way you can do that, know for a fact, is by genetic testing, then the sibling recurrence rate is probably close to 0, okay? So in other words, is if you genetically test the kid and the parents, the kid has a mutation, both parents do not, you can tell the parents that the chance of having another kid is close to 0. I think it's extremely unlikely. So that's important information. That being said because the child has a dominant mutation, the chance that the child will have another child, in other words, the offspring of that child, the risk is 50%. 50%. So genetic testing can dramatically change the perceived odds of what the baby has. So that's why I think that it's important to do. Okay? So, which patients should undergo, or why should patients undergo genetic testing? One is as I said, there are implications because they change so dramatically for both sibling recurrence and recurrence for future generations. You could also, there may be a need for additional screening of family members. For instance, if you do have a gene associated with like Notch1 in a child who has hypoplastic left heart syndrome, and the parents also have that mutation, then they should be screened for a bicuspid aortic valve or something like that. There probably is a higher incidence of bicuspid aortic valve, which obviously could lead to a thoracic aneurysm later in life, or they may be prone to AS, AI later in life, okay? There's also potential for other organ involvement. For instance, if you know that this tetralogy is associated with JAG1, then you know he's got allegeals, that patient is going to have liver problems. So it can inform you about some of the potential complications that that child has. And we're working on some studies now that the genes can have some implication for clinical outcomes. For instance, one of the most common mutations that we do see is associated with chromatin remodeling genes. And chromatin remodeling genes in patients who have some severe forms of congenital heart disease have a 35% of them will have neurodevelopmental abnormalities. So there are some implications for clinical outcomes. So if you have suspected chromosome anomalies, except really for trisomy 21 because it's so, but if you have a syndromic patient, I test all of these people. If you have interrupted aortic arch or truncus, I test all these people, particularly for DeGeorge. And because conal truncal anomalies have such a high incidence of DeGeorge syndrome, I actually test essentially all conal truncal anomalies. Branching, these are also associated with DeGeorge, and alveolar tract also. Mind you, I've made it sound like everybody has a lesion. It's not true. Actually, when you genetically test someone, you're actually unlikely. You are less likely to find a genetic cause, right? If it's when you do find a genetic cause that it may have some potential meaning, but that's just, actually the most likely thing that you'll find is that there's no genetic lesion. For instance, a Denovo dominant mutation in a congenital heart disease patient, a complex, it's only 10%, okay? So just be aware of the general numbers. And really, if you have an isolated simple CHD like a VSD, I never test them. Your diagnostic yield is going to be close to zero. And actually, despite everything, they've done a lot of genetic testing on heterotaxis, and the diagnostic yield is very low. It's surprising. Okay, so there are many different genetic tests. I'm just going to go through them quickly. Karyotyping is the old gold standard. It can only detect large chromosomal anomalies. I don't get it. FISH is basically DeGeorge and Williams. Some people get them, especially if there's more clinical suspicion. But I usually go straight for a chromosomal microarray. It identifies these middle numbers, CNVs. You're going to miss 10% of the sequencing, but it's the most bang for your buck. We have in the United States targeted sequencing panels for very common gene mutations. You can get those. All of the research that we've been doing lately is basically on whole exome sequencing, which is the exact sequencing code of the genes that produce proteins. It only represents 1% of the genome. Actually, when we started 6 years ago, well, 15 years ago, the cost of sequencing of the exome was I think it was $1 billion. But currently, to sequence the exome is around $300. But even still, clinically, they'll charge you a lot more. That's kind of like the research price. Okay, what test should be ordered? Chromosomal microarray. It'll detect in overfish. It's about probably 10% better, and that's pretty much it. I wouldn't necessarily suggest whole exome or whole genome sequencing. Okay. All right. So in conclusion, we've covered like a lot of stuff, but here it is. Okay. The mutations that lead to congenital heart disease come in 3 varieties, right? Each contribute about 10% to our total knowledge. The big ones are chromosomes, chromosomal aneuploidies. Aneuploidy just means an altered number, different number of chromosomes. Middle ones, which are copy number variants, and very small individual ones that are called single nucleotide variants, so 10, 10, 10. They cause problems by causing an altered gene dosage of developmental regulatory proteins, not structural proteins. Sibling recurrence is around 3%. Offspring recurrence is 5 to 10%. For fathers, it's around 5%. For daughters, it's around 10%, or mothers, it's around 10%. LVOTO may have higher. The gene test to get is a CMA, but they're going to miss some things. And when, especially in the States, they're going to offer whole exome sequencing, I would just say no. And that's it. Thank you very much.